Good morning, everyone. It's time to get started. Thank you for joining us. My name is Steve Medevanga. I'm the head of biological sciences and also molecular cellular biosciences. Thank you for coming. Just a couple housekeeping announcements. This is a hybrid event. We have people on Zoom. How many people?
40 so far and joining. And Steve, I'll ask you to just move a little bit closer to the podium.
Okay, now the Zoom people can see me. About 40 people on Zoom. I'm going to actually take this off just momentarily. This event is being recorded. I will also say that there's a sensitive microphone system overhead, so if you could please keep chatter to a minimum or eliminate it completely, otherwise you'll be picked up by the microphones. Please, if you could check your cell phones and out of respect for our speakers, turn them off and silence them if you could, please. Following our talk today, we will have a question and answer session, and remote participants are encouraged to submit their questions via the chat box. Before I announce the speaker today, I want to talk about a couple guests that we have in the audience with us. First and foremost is Miss Isabellita Abelle. She is the Chief Executive Officer of the U.S. Lumber Company here in South Jersey. She has been a member of the Rowan University Board of Trustees since 2012. Lita, thank you for your service over the years and being such a great steward to our university. If you could please stand and accept our appreciation. I also need to mention Dean Vojislava Poplistic from the College of Science and Mathematics. She is our host for today's event. Boki, thank you for sponsoring this. We could not have pulled this all together on short notice without Bridget alone, and Bridget does not want me to mention her, but Bridget, please accept our appreciation. And now it is my pleasure to introduce to you Dr. Rhodora Romero-Aldmetia. Dr. Aldmetia earned her Bachelor of Science and Master of Science degree at the University of the Philippines. She then came to the United States to earn a PhD in botany from Purdue University, working with Professor Thomas Hodges. After that, she traveled to Germany for her first post as a postdoctoral fellow at Albert Ludwigs University, and she became a member of what is called the Golden Rice Project at that time. She has had quite a number of former positions. I only have time to mention a few. She was a research scientist at the International Rice Research Institute in the Philippines. She was the Chief Science Research Specialist at the Philippines Institute. She was the president of the Philippine Society of Biochemistry and Biology. She was also the former president of the Philippine Crop Science Society, and several others as well that I don't have time to mention. She currently is the editor-in-chief of the Philippine Journal of Crop Science. Her awards are numerous. I will mention just a few. She has earned the 10 Outstanding Women in the Nation's Service Award from the Philippines. She is one of 10 outstanding young scientists recognized in the Philippines. She won the Science Prize in Biology from the National Academy of Sciences and Technology, and she won a similar award from the World Academy of Science in Italy. She has numerous publications that she started as a bench scientist in plant biotechnology. I'll mention just one of them right now. The title of this talk is Agribacterium tumefaciens Mediated Transformation of Japonica and Indica Varieties of Rice, Oryza sativa. She promises me that she'll keep the technology down a little bit during our talk so we can all follow along. I remember when I was a doctoral student and we were studying plant biotechnology, and we learned at the time how difficult it was to transform monocots, grasses, and most of our crop plants are monocots. Indeed, rice is the most important crop plant in the world. It feeds 50% or more of all humanity. And Dr. Aldmetia was a pioneer in the transformation of rice. She did it for her PhD work and continued at some of these international institutes later on. Her technologies that she developed as a doctoral student are still utilized today. Her very famous publication that I mentioned is still cited to this day. We're extraordinarily pleased, and please give a warm welcome to Dr. Romero-Aldmetia. The title of her talk is Biotechnology Innovations for Food Security and Environmental Sustainability in the Changing Times.
Good morning, everyone. This is my second time that I'm here at Rowan University. The first time was in 2017. 2018, I can't remember. But I see that there's a lot of enthusiasm as we gather a lot of students here. I'm very happy for the invitation. Of course, it's through my aunt, my aunt Isabellita Abelle, and of course, our dean, Dr. Boki, and Steve, and Bridget, and where's Joanne? She's aware. But I'm happy to be here and to give you a presentation. This presentation, it has been, I have delivered this talk in Southeast Asia. Recently, I gave this talk in China through a hybrid system. And of course, I was able to, well, they asked me to translate it, to give this slide to them early on so that they can translate it into Chinese. And they published it, and now they wanted the narrative. So I made a narrative of 8 pages of this one. It's also translated in Chinese. That's how my work is. So I'm currently the director of the ISAAA Southeast Asia Center, and at the same time, the Global Knowledge Center on Crop Biotechnology. So if you have your internet or your Wi-Fi, you can see ISAAA.org. That's our website, and we have a lot of materials there for consumption of the students in layman language, and targeted to different stakeholders, from policymakers to students to youngsters, farmers also. It depends on what interests you or what you are in that stakeholder. And also we have, so what I'm going to present to you is a data set on the global adoption of phytocrops. So ISAAA has been compiling that data since 1996, and the latest one was 2019. But because of the pandemic, it was very difficult to obtain the 2020 data, but we still have to write that for publication, perhaps in the first quarter of 2022. And also, I'm going to present to you the new innovations, which is the genome editing. So for now, so that I will know how I will tackle the discussion, may I know who knows about biotechnology or genetic engineering? Okay. How about genome editing? Wonderful. So this is related to medicine or healthcare, is that right? Right. So my work now, the discussion I'll do today is for food security and environmental sustainability. So it may not be what you want to hear with regards to health or relation to health, but of course it will help you with how you discuss to your students and how you understand what's going on currently. Okay, so why do we need phytocrops? Because of high global population, we know that. We have currently, we are currently 7.9 billion, 2019-2020 data. And then, so it's estimated that we'll have this one and we'll have that population by those number of years, and we need 70% increase in food production. And this is a lot of challenge for our food producers. On top of that, we have climate change. We are experiencing it right now. So there are farmers who do not know when to plant anymore because the patterns of rainfall and long and short day length has changed during this climate change. So, and it also affects food production. And there are new challenges like biotic challenges. So what are they? So there are plant pests, diseases, viral, bacterial, fungal, and also insects. At the same time, we have abiotic stresses. So we have submergence, and then the opposite, which is drought. So with climate change, all of these are coming in. We have felt this in the United States. We have felt this in 2012, when you had a big drought in 2012. And that was when corn was, there was a shortage of corn because there wasn't so much planting. It was all devastated by drought. So from that time on, we have experienced this all over the world. So drought has been affecting Australia, Latin America, and of course, Africa. So it has been the interplay between high global population and the climate change will be putting a lot of stress on food insecurity, especially in the low-income countries. So with more than 820 million still hungry, the 2015 Millennium Development Goals of the United Nations was not a success. You know that, right? Now we have to embark into SDG 2 goal of zero hunger by 2030. And this will require biotechnology applications, because if we do the business as usual food production, we're not going to feed these people, this number of people. So with that, that is an overview of what the challenges, what food producers are facing in terms of challenges. So our plant producers have been doing traditional breeding. You know that traditional breeding, when you have the male-female part of the flower, or the male-female crops or plants hybridized, and you have the new varieties. But then by the year 1970s, we had this mutagenesis. Anybody who knows mutagenesis? Of course, you know that. And then we have chemical irradiation. And since the 1970s, we have a lot of crops, especially staple crops, rice, wheat, corn, which have been developed through mutagenesis. But then it's still random, and you don't know where the genes go, or which part of the genes have been mutated. So then scientists decided to do transgenics. This is where you have, okay, I will just put that the discussion later. We had transgenics, where we have recombinant DNA methodology, and then we had genome editing. This is the newest one, where we have the Nobel laureate awardees last year were discoverers of genome editing tools, the CRISPR. So this is how we develop the technology in order to respond to the challenges of the times. And so now let's look at the basis of biotechnology. So we know about this, the DNA having the pairings, and every organisms on Earth have this. Of course, there are unique sequences that make organisms different from one another. So now we have crops, we have animals which have altered or modified or improved traits because of biotechnology. So let's see how we do that. In crops, my expertise is on rice, because I'm a rice biotechnologist at the International Rice Research Institute before. So what I was doing before was to get a gene, put it in a gene construct, target to the cells through agribacterium pluripotent, or through particle bombardment. Oh, you know this, right? And then you select the colors, you generate into plants, test in the greenhouse, test in the field, and then after some time you have a variety. It's simple, because scientists have to go through regulation. Each country that conduct genetic engineering research have to go through regulation. From the time that you get the gene, to the time when you get the gene construct, and do every step of the way, you have to undergo regulation. So when I was a student at Purdue, I went home to the Philippines and started my lab, and I have to go through all this. And it took me a year before I can do work in my laboratory, because I have to be checked. The laboratory that I'm working in has to be checked on conformity with the rules. And that is also the same way with other countries. During the time, well, I was a Rockefeller Foundation scholar, and most of us were from Asia. A lot of Indians, Chinese, Filipinos went to the United States to study, but upon going home, they have to undergo this exercise. They have to establish a lab and establish also the regulations. And then, so after all those years, from 1996 up to now, we have different applications of biotechnology. So we have been fighting diseases. So we also already have new biotech products, just like insulin, vaccines, which were produced through biotechnology. And you already know about the vaccines, COVID vaccines and vaccines against diarrhea and many others. And then in increasing yield, our scientists have already improved our crops through genetic engineering to increase the yield, environmental friendly so that it will not be polluting our environment. Safer crops, because we do not need anymore to spray hazardous chemicals to kill pests and to kill the weeds as well. So safer crops, nutritious food. And I'm happy to tell you that since I'm a member of the Golden Rice Project, the Philippines has been given an approval for commercialization of golden rice just in June. So we're very happy that we now see the fruit of our labor. I did my postdoc for golden rice, and now it's going to be in the market very soon. And better industry because we have more yield, so we have more raw materials for our industry to move. So these are the potentials of biotechnology, and we see that in our everyday life. Now moving forward, so looking at the statistics. So ISA has been monitoring the area planted to buy the crops. So you can see here we have started monitoring it since 1992. But the modern one, or the commercialization one, started in 1996. And we see that the developing countries started very late or very, very slowly, but the industrial countries have started immediately. But then in 2011, you see that there are now more developing countries that have adopted biotech crops compared to the industrial countries. And you see some of the ups and downs in all this, especially there, is because of the price, demand of the commodity. And we are looking here at the soybean and the corn demand, okay? So we're happy to tell you now that developing countries are getting into biotech crops because they know the benefits that they can get from biotech crops. I'll mention that to you later on. So this is now the countries which have planted biotech crops. So we have the top 10. Of course, the US has the largest area planted to them, and followed by Brazil, Argentina, etc. You can see that. And then we have 3 new countries, 6 African countries. So we have now Ethiopia, Nigeria, and Malawi, which have planted biotech cotton in 2019. So the Northern Americas with the US and Canada had 44.1%. The Latin America had similar, and then Asia Pacific follows, and Africa and Europe. So we have only 2 countries planting biotech crops in Europe, which are Portugal and Spain. And this is only the BT corn because they have the European corn borer, which is very, very devastating in their parts of the world. So there was a slight decrease of 0.7% from 2018 to 2019. And this is because of climate change, the drought. Right. So then let's move into the top 5 countries are the US, Brazil, Argentina, Canada, and India. And at this, we know that there are 2 industrious countries, 3 developing countries, right? And the adoption rate is very close to saturation, meaning very close to 100%. So these are for biotech crops: corn, canola, soybean, and cotton. And Argentina has the highest adoption rate, very close to 100% in these crops. And so these are now the crops that are planted over the world. So the Northern Americas, which has USA and Canada, we planted. We have, you can see there maize, soybeans, cotton, canola, sugar beets, alfalfa, papaya, squash, potato, and apples. So people here from many different parts of the world, you can see what crops are planted in your countries. I'm from Asia Pacific, so I'm from the Philippines, so we're only planting stock trade maize or corn. And of course, Europe, maize. And oh, I cannot see the lower part. There is another country here, Malawi, Nigeria, and Ethiopia, and they're planting cotton. And Nigeria is very significant because they are now planting the cowpea, cowpea, food crop, compared to the other countries which are planting cotton. In terms of crop, you see that Biotech soybeans has the highest. Why is that so? You know, because it's used for human food and also used for livestock, followed by maize, and then cotton and canola. Canola is for oil, and of course, cotton is for whatever we wear. Now, you can see that Biotech soybeans has also gone up and down. It depends on the climate change, the demands of the livestock and poultry industry, and of course, competition, because of course, the Americas is competing, the Northern Americas are competing with the Latin America, so they have to balance out. So that's not going to flood the market as well. Then globally, so this is what we're looking for. Globally, we have 79% of the whole cotton already GM. And I think this is 76% for 74% for soybeans, 31% for maize, and 27% for canola. So you can see that we still have a lot of room for corn. And corn is a staple crop for humans and animals, also for livestock. So countries, well, the US has a high GM corn adoption, but in countries of Africa and Asia Pacific, we still have a lot of room to plant GM maize, especially in China and in India. Those are big countries with high population rates. So we are into advocacy or telling them that if you go through genetic engineering or biotech crops in corn, it could somehow solve malnutrition, and it could help you feed your country. And in South Africa, we have white corn, GM white corn for food, for human food. But most of the GM corn across the world are for livestock. Well, we have yellow corn. You know that your yellow corn here, sweet yellow corn, they are GM. You know that? Yes. So it's very good to know that this is varying. It depends on the price. So this year, I'm looking at the 2020 data, there is a shift. Sometimes soybean has the highest adoption, sometimes it's cotton, so they interchange. But last year, in 2019, the cost of cotton in the market was high, so there were more farmers who planted cotton, so the adoption rate was high. I included this because I'm from the Asia Pacific, but just for you guys who are interested. So there are 9 countries which were planting biotech crops in Asia Pacific. So we have China, Vietnam, Philippines, Indonesia, Australia, Myanmar, Bangladesh, India, Pakistan, and of course, importing countries. What do you mean by that? So they do not plant, but they import, just like Singapore. Singapore doesn't have a large area to plant. They don't have agriculture, but they have money to buy, so they import biotech crops the same way with Japan. Japan and South Korea, those are big importers of biotech crops because their agriculture cannot embrace biotech crops as yet. Great. We can see that herbicide tolerance was the highest. It went up immediately. This is because of soybean. Because soybean is very much in demand for livestock and also for human consumption. And then we can see that BT, or the insect resistance one, started also But as we go through the years, we can see that the stock rates have gone up. And because farmers, our farmers are now learning the technology, they want to save on money so that they don't have to spray herbicide, or they can use a much safer herbicide, which is glyphosate, and then they don't need to spray insecticide. So by the data, by 2019, we see that the stock rates, the areas planted to stock rates has gone up, and it has surpassed the herbicide tolerance area. Now, looking at the new additions, so we have, these are the existing adoption of biotech food and feed crops. So I have mentioned about the 4 major crops. So we have now, we know this, the non-browning potato, which is already available in the US and in Canada. It's now planted at this hectarage. And non-browning apples, it's already commercialized in the US and Canada as well. And we have eggplant. Anybody who eats eggplant here? Well, in Asia, it's a staple. So it is planted in Bangladesh currently, and it has gone up to 1,131 hectares. And my company is also a member of the eggplant advocacy. So we have just received an approval for propagation, for food, feed, and processing as yet. Because in our Philippine regulation, you have to be approved for food, feed, processing before you can be approved for propagation, because we have to know that it is safe for human consumption. But it's already being used in Bangladesh since 2014. And they have already reduced the number of sprays to the eggplant because they are already insect resistant. So why spray, right? So they have reduced the exposure of the farmers to the insecticide, and also have increased the yield and the acceptance of the Bengali people. And also we have the Harb Extra Low Lignin Alfalfa. This is for our livestock, because we know that alfalfa has a lot of lignin and it's not digested by our livestock efficiently. So it has low lignin, and so the manure of our livestock will not be difficult to decompose. So there is digestibility and reduction of pollutant in the environment. And it's already adopted in Canada and Argentina. We have insect-resistant sugarcane. This is also a new one now in Brazil, drought-tolerant sugarcane in Indonesia, and a high lake safflower in R&D, but it's already in the market in 2020 in Australia. And we have the pink pineapple. Have you ever seen a pink pineapple in the Philippines? You have, yeah, you have. And it's high anthocyanin and very sweet. It started, of course, the technology was developed in Australia, but this was bought by Del Monte, and then they have a seed increase in Costa Rica. So it's now going to be commercialized very soon. I think it reached the Philippines this year. So it's very, very attractive if you put it in your cocktails. Yeah, because it's, but it's very nice because it has high anthocyanin and very sweet. They said, I haven't tasted it. And it is increasing also in hectarage. Oh, okay. So now those are what we are having in terms of what's in the field, what's commercialized. So we're compiling, we have a GM approval database at our website, and we have put together data on approvals. So from 1992, because we have done that from 1992 to 2019, we have accumulated the registry in different countries all over the world. And there are 72 countries already issuing approval for 4,485 approvals for 32 GM crops. And the US has the most number. Corn has the largest number of approved events. And herbicide tolerant maize has the most number of approvals. This was the data for 2019. So if you want to look at the website of at our website, you go to the GM approval database and you get the latest additions. We update as soon as an approval comes. And what do we get the approvals? Of course, we contact the developers, or the developers contact us that they have already issued approval for food, feed, and processing, and for propagation. Currently, the ISA GM approval database has a more updated data compared to the Cartagena Protocol on Biosafety, the CPB, Biosafety Clearinghouse. And so scientists go to our website before they go to the clearinghouse, because that one is not updated as much. So we're maintaining this one. And from that, we have seen that these are the new approvals in 2019, mostly from the US. Drought tolerant cotton, first one. You see, this is a response to climate change. Low gossipol in cotton. The scientist was from Purdue University, Philip Tor, a very, very good friend. And gossipol is the chemical that is in cotton. And you know, cotton seed, when they process the cotton seed is left. So what they do is they feed it to livestock, but it has gossipol that makes abortion, or it affects the livestock. So they did genetic engineering of that, and they lowered down the gossipol content. So it's now approved for the market. We have non-browning apple for another Fuji variety. Polyunsaturated fatty acids in canola, multi-insect resistant in Brazil, Argentina, and so on and so forth. There are still more, because they are coming one after another, and the 2020 data is full. It's very good data. And so what do we have in the pipeline? Non-browning romaine layers, you like this, right? Colorado beetle resistant potato. This is a drought tolerant wheat approved in Argentina for propagation already. And nutrient use efficient, water use efficient, and salt tolerant rice. This is a research done with some US universities and also with some African scientists. So it targets a lot of traits, and it's on the staple rice. So let's move to switch gears for a moment. And we can see that due to, this is for crop yield on corn, we can see that because of molecular breeding and biotech, we can see that the average corn yield has gone up. If we are going to go this way, it's not going to be as sloping. It's almost negative sloping. But when you see through molecular breeding and biotechnology, the yield is really going to the positive slope. So we know that when we are looking at 2030, we hope to hit that mark when the SDG 2 goal is reached. That's why the title of my talk is, in these changing times, we have to do something. Otherwise, everybody will go hungry. So with that, I'd like to give to you some of the benefits of Biotic crops. This is now based on GM. I'll be going into genome editing in a little while, but you see that there is increase in crop productivity. We conserve biodiversity because we do not need to denude our forests or cut down trees, and so that we can increase production. So we can save this 1 million hectares of land. We have a better environment because we save 776 million kilograms of, there's a global reduction in the use of crop protection products, which are mostly chemicals. We can reduce carbon dioxide emission, and most of all, we can alleviate poverty and hunger. When we were talking to farmers, they're now more contented, or they're now more restful when they sleep, because they know that the following morning they can still see their crops without worms. They can see their crops flowering and bearing fruits because of biotechnology. And that is something for farmers. Now, moving on to the new innovations, which is genome editing. So we have seen how plant breeding works, how genetic engineering works. Now we know that genome editing has come beginning in the 2020s, 2000s. So we have, what is this? So with the activate, it's that we either deactivate an unfavorable characteristic or a trait, or we enable a beneficial character. How do we do that? So we can do this through genome editing or gene editing. For some scientists who I am in contact with, they want to use gene editing. Some people want to use genome editing, but this one is gene editing, so I'll just use that. This is from an ISAAA slide. So it's a technique that allows breeders to make targeted improvements. Targeted, because before in genetic engineering, it's random. You don't know where the genes get inserted. But you have to have a lot of regenerants or genetically engineered plants so that you can do some evaluation whether you got the gene inserted. And then you will see where they got inserted. But in genome editing, you already know where they're going to be inserted. So it's targeted. And the expression also, because you know that as soon as it gets there, you know that's going to be expressed. Gene editing tools have been developed. We have CRISPR-Cas9, which integrate desirable traits into improved seed products with greater efficiency and precision than before. And also efficiency in terms of time, and time in the laboratory, time from the laboratory to the market. And also precision, because you know where it is going to. And also, it does not go through very difficult regulation. This is one thing that genetic engineers have to work on. As soon as they have a genetically engineered crop, they have to go through a series of regulation, which is more expensive than the laboratory work. And that makes it, it gives a negative impact to our scientists. So most of the people who are doing genetic engineering are moving into genome editing. But it's not that we have to get rid of genetic engineering. It is still useful. And it's still very useful for multiple traits because you cannot do gene editing in multiple traits. So genome editing and genetic engineering are staying. And that's a good idea. Okay, so how do we do that? So it's similar to a word processor, if you know the word function, the word function in your computers. So in your app. So if you want to change something, it will from C to T, or what is it? From C to an X or something that you can change. So it will be the function of the gene can either be deletion or loss of function, or it can be a replacement in function, okay? And I'll give you some examples. Okay, so these are the ways how to do it, 3 ways. You enable a beneficial characteristic or drought tolerance. It's the activating because you have an unfavorable characteristic being deleted, and you can break genetic linkages, which is important for some traits. So these are very important traits in the new times where we have climate change and the development of new plant diseases and pests. How is my time? Okay, I have to go fast. So potential benefits in agriculture could be in farmers, consumers, and the planet. And you can see the potential applications in many different traits. Weed management, this is resistance, drought tolerance, quality improvements, and enhanced nutrition. And we already have this in the market. This is the first genetic genome edited crop. and product in the market by Calyxt. And so this is a soybean with high oleic acid, so it's in oil. It's a nutritional trait for consumers. And this one is now from Japan, which is what we call GABA tomato, because they have the glutamate decarboxylase key enzyme, which the key enzyme for making this amino butyric acid. And we can see that through the deletion of the C term, there are 2 genes of the GAD, we can increase the GABA. And that enhances blood pressure lowering. Actually, this is already commercialized in Japan, and there are already in the markets, in the anywhere. So it's very useful. And I hope they're going to export it to some other countries. So these are just examples of how genome editing works. So if you, the number of edited genes, for example, in wheat, will make you having, if you deleted some of those genes that limit wheat seed size. Another one is, okay, so it increases the number of edited genes. So you see that the grains are increasing as well. So this is an example from the International Rice Research Institute, where we have the deletion of genes responsible for limiting the weight, the grain weight and the grain number. You can see that if you have deleted the most number of genes that governs or that limits weight, grain weight and grain number, you see that there are more grains and more weight. This is for resistance also in rice at the International Rice Research Institute. So they have deleted the receptor gene of the BLB, so that when a bacterial leaf blight pathogen attacks these rices, they do not have the receptors anymore. So the bacteria cannot attach to those receptors, causing diseases. So you can see that these are now the genome edited rices. And also for fungal resistance, another one, rice from the International Rice Research Institute. And also for amylose content, for beta carotene. Okay, so this, you know that the lycopene and the okay, backtrack. The lycopene and the carotene are in one metabolic pathway, so that when you have high lycopene, you don't have carotene. When you have carotene, you don't have lycopene. So what the scientists did is they deleted this gene that converts lycopene to carotene. Now we have tomatoes that have high lycopene content, although they are not as red. But the lycopene is very high. Why is lycopene important? It's for your antioxidants, right? Okay, so that's one. And then for phytate, okay, you know that in livestock or in piggery, phytase is very important because some of the nutrients that are involved in phosphorus uptake are chelated. So our plants cannot get the ionic form of phosphorus because it's bound. Now, because of phytase, if it's in the crop, the phosphorus is already made available. So the phytase gene is removed. And so now we can have a phytate gene, more nutrients that are available for our livestock. Okay. All right. So those are just few examples. There are so many examples, and across the world, we have regulations for genetic engineering. Now, people are looking at genome editing not to be under genetic engineering because they're like mutations, because you're just deleting one one C or G, one nucleotide, and you can just put them together. So it's just like it happens normally in nature. So are you familiar with SDN1, SDN2? Okay, that's a little bit technical, but suffice it to say that currently more countries are getting into policy considerations that genome edited crops should not undergo genetic engineering regulations as long as the new product is available in nature. It just so happens that genome editing can do it faster. So if there are questions on regulation, I can help you with that, but if I dwell on it, it's going to be another lecture. So currently, this is what we need to do. We have to be diligent. We know what the science is. We know that there are a lot of people who are against genetic engineering and biotechnology as a whole. But because of the potentials, the benefits that they can bring to the whole global population, we need to be diligent and help in forward-looking regulatory steps based on science. So this is where, when I say that our policy considerations should always be based on science, not based on traditions, not based on ethicals, and so forth. So we have to also look at the benefits instead of the risks. This is one way where you can do science communication. I'm going to give a lecture on science communication, and we have to look at the benefits. You can say that the risks can be managed, but we have to look at the benefits first, and then the risks, because the risks can be managed. And agricultural productivity with environmental conservation and sustainability is the key to feed our whole global population. And we have to consider millions of hungry population. We know that due to the pandemic, this became more rampant. I haven't seen the data, the United Nations data on the effect of COVID-19 on hunger. I think it's going to shift the SDG goals again of the United Nations because of this pandemic. So with that, I thank you very much. This is our last slide. Biotechnology continues to be important to meet the 50% increase in food demand in 2050. This is a publication way back in 2018, but now this has changed to 70%. We have to increase to 70%. So this is our ISAAA website. You can look at that while we're doing the Q&A. This is the pre-55 global status of commercialized biotech GM crops in 2019. I'm still putting together the 2020 data, and people are running after me, but I haven't put that down yet, but we hope to publish that by the first quarter of the year next year. But we have an upcoming publication on primer. It's a primer on new breeding technologies. It's going to be at the ISAAA website by the end of December. And we have invited experts on genome editing, the science and potentials for crops, for animals, the regulation that governs it, prospects in Asia, prospects in Africa, how it can be communicated, or how can we advocate or science communicate genome editing. And the last one would be how genome editing can contribute to food sustainability. Yeah, thank you so much for your attention.
Thanks so much. We have about 10 minutes for questions for Dr. Aldmetia. I think we can have a couple questions from the in-person audience. We'd like to also have some questions from our remote audience as well. So any questions from the audience?
Yes, ma'am.
Hi, thank you again for your presentation. I know you mentioned very early on just talking about like CRISPR. Do you think regulations are the biggest kind of like pushback on why CRISPR is not being used more in the food industry?
Yeah, I think so, because when biotechnology started, or when commercialization of biotech crops started in 1996, we were hoping, well, the developers were hoping that acceptance would be immediate. But then there were a lot of skeptics, especially the other side of the fence. And so we have to, and they asked for regulation. Actually, the regulation came up because of, they were saying that there are risks. So each country was then asked to provide that. And if you're familiar with the COP, Conference of the Parties, meeting of the parties of the biosafety, Conference of the Parties for Biodiversity and Biosafety. And this is where there is a meeting of the minds of the anti and the developers. So this is where each of the countries that are now doing or conducting biotech crop research should undergo risk assessment. And that is it became the segue to having regulations. And actually it is a key, how do you say, it's the barrier, it's the key, and it's the barrier to commercialization in a certain country. But there are countries who were able to propagate or adopt biotech crops without regulation. So for example, in Myanmar, Myanmar has been propagating biotech cotton. When we discovered it, they were propagating it since 2010, and we discovered it in 2018. And it was doing very well without any regulation. Because acceptance was there, they understood the science. But because of the other people were able to know that, oh, they're doing it without regulation, they have to do it, like that. So as ISA, I went to Myanmar and we helped them with the support of the USDA. We were able to help Myanmar do their national biosafety framework. We have this political instability in February, so we were stopped. But the NBF was already there. It just needs a signature and a launch, but we were stopped. And then we also have those countries where, for example, Vietnam. Vietnam was doing field trials without regulation. And as soon as they had the regulation in place, Vietnam was able to adapt to biotech maize, and they have started planting in 2014, and now they are increasing. And then we have also political will, just like in Bangladesh. In Bangladesh, the regulation was there, but the political will was very strong. The secretary of the Department of Agriculture really endorsed biotech crops, and she even handed BT eggplant seedlings to the farmers. So that's how regulation and political will, when they are together in a country, adoption is very smooth. Yeah, thank you.
I'd like to offer a question from online since we have quite a few. You discussed how important GMO crops are for helping farmers in developing countries. How do farmers in developing countries acquire genetically modified seeds that may be patented or have licensing fees?
Oh, okay. Actually, the cost of the seeds already include those licensing fees, and because of course, especially for biotech crops which were developed by the MNCs, but for locally developed biotech crops, it's not there. So the cost of the seed is lower, and it's there's no the licensing is not there because, for example, the BT eggplant. This is another example. Yeah. So in Bangladesh, the crop was developed as a humanitarian act by Monsanto to give to Bangladesh. There was no profit involved because they want to help. So the Bangladeshi were able to put the gene from the variety where the GM variety to their local varieties. So they have no proprietary, no licensing involved, and the seeds are given for free because it's not the hybrid one. It's the open pollinated or the open pollinated, sexually propagated seeds for BT eggplant. So there's no proprietary there. But for GM corn, for soybean, which are developed by the MNCs, the cost of the seeds already include the IPR of the technology.
If you could take the in-person question.
So speaking of the BT eggplant, BT corn,
Mhm.
has there been any observation of resistance development in insect populations to become more resistant to the BT toxin in those
countries? Yeah, of course, scientifically it can happen, but our technologists, technology developers have put together a system. They call it the stewardship, so that the resistance will not break down. For example, we have in one bag, we have GM and non-GM seeds in it, so that the insects will not develop the resistance to the insecticide. So that can be managed. That's why when I'm telling you about the risk, all of the risks that come up can be managed. And so this is insect management technology. And speaking from the Philippines, we have an IRM, or Insect Resistance Management Committee, which takes charge of all the MNC's approval if their bags of seeds can be sold to the farmers if they have enough GM and non-GM seeds in it. For the question.
So similar along those lines, with respect to herbicides, are you concerned that herbicide tolerant GMOs lead to increasing use of more herbicides and the development of super weeds?
Okay, that has been asked very, very frequently. You know, that also happens in nature, and our scientists just cannot do away with all this skeptics. So they have to devise a way in order that resistance to the herbicide will not develop. So they can put several, the stacking of genes. They can stack different kind of genes in the seeds, so that, for example, in this season, we'll be using dicamba. In this season, we will be using 2,4-D. So there's a stacking of a gene, so that in each season, the herbicide that they will use will differ from one season to another. So the development of how do you call this? Weed resistant crops will not happen. Yes, sir.
You mentioned the Golden Rice Project. I wonder if you could talk just a little bit about that and also its relationship to vitamin A deficiency in humans.
All right, thank you so much for the question. Actually, that is part of my postdoctoral in Germany. Okay, so we know that vitamin A deficiency is a problem in developing countries. And how does it affect the poor, the people who cannot access vitamin A, the eyesight, the susceptibility to infectious diseases, and also for youngsters being not able to develop mentally. And so those are the vitamin A deficiency, that the hidden problem of vitamin A deficiency. Now, in Germany, it was started in Zurich when vitamin A gene, the gene responsible for the development of beta carotene in rice, was obtained from grass, the daffodil, the yellow daffodil, and so it incorporated in there. And then they got another gene from a bacteria, and so they were able to develop beta carotene enriched rice in a variety of rice, which is a japonica, the Japanese rice, which is round. But then it wasn't enough. I mean, the RDA, or the recommended daily allowance of vitamin A, is not enough in that small one, from that genes, from daffodil and from the bacteria. So what happens is, in 2004, scientists from Syngenta used a gene from corn, and then the bacterial gene. So the same bacterial gene plus the gene from corn. So the previous one was daffodil and bacteria, which did not give much beta carotene, and now they shifted to a source, a corn source, and the bacteria. Now they have improved the amount of beta carotene in it. So what has happened is when we take in beta carotene enriched food, so for example, you eat a lot of squash or carrots or anything that has high beta carotene, your body converts it to vitamin A. So this new variety of rice had more beta carotene in it. I think it's 37 times more than the previous one. So they got the line, I think it's line G, golden rice line G, and they incorporated it into rice varieties that are being cultivated in Asia, where there is a rampant vitamin A deficiency. So we targeted Bangladesh, Philippines, and Indonesia. So the Philippines had the most advanced because the International Rice Research Institute is there. So the project was launched at ISAAA, and then Bangladesh followed suit, and then Indonesia. So currently, we have this, the genes have been introgressed from the Japonica rice to the cultivated one. So we have several lines of indica rices, which can now be cultivated and can be consumed. So last I think it was only at the end of October when they launched this. So there were several people in front, and they have boiled golden rice. They took pictures eating the rice, and they were asked how it tastes. And I was texting with my friend, a USDA, US Embassy Manila. And I asked him, why did you finish all the bowl of rice? Because they were given each a bowl of golden rice. And he said, it's so tasty, I ate it all. And he said, because during the picture taking, everybody was showing the bowl of rice and his was empty. And he told me that it doesn't taste different from our usual rice. And it tastes good also. So it's a good thing that it's already launched. It's going to be available to us. And of course, the developing countries is the target of this golden rice. So that's how it is.
Yeah, so we're just about at our time limit. So I have one more quick question. So what proportion of GMO crops are consumed directly by humans versus used for other applications such as feeding livestock?
Okay. Most of our biotech crops, especially I cannot say the percentage because I do not have the data for that. But I'm confident that corn and soybeans are both being used for livestock and food. Because for our everyday purposes, just like we have soybean oil. Are you aware of that? We have soybean oil, which are GMs. We have canola oils that are GM also. And for cotton, of course, we don't eat cotton anyway. We're using it for fiber. But of course, there are countries who use cottonseed oil. And so, for example, in India, cottonseed oil is very much used because they do not have much canola there. So they're using cottonseed oil, as well as in China. And so I may say that we are using it 50-50, sort of. Yeah. Thank you.
We need to wrap it up here. Before we leave, I want to announce that there'll be another talk by Dr. Romero-Aldmetia in this room at 3:30 p.m. today, Raising Public Consumer Understanding and Perception of Ag-Biotechnologies. Immediately after that, there'll be a brief reception. Yeah. In case you'd like to meet Dr. Romero-Aldmetia or our trustee Abelle. So please come back for the afternoon session, and let's give a warm thank you to our speakers.